Thank you, Dr. Prokop. And I think we heard that last message loud and clear, that it's a complex process to get the drugs on the market, including both the technology and the systems, the several different soft systems processes. Our next speaker is Dr. Jim Leary. Jim is going to speak on the topic of programmable nanoparticles for drug gene delivery in regenerative nanomedicine. For everyone's background, Dr. Leary is an endowed professor of nanomedicine and tenured professor in the Weldon School of Biomedical Engineering at Purdue University. He is also a member of the Burke Nanotechnology Center and the Bindley Biosciences Center. Recently, Dr. O'Leary was elected Fellow of the American Institute for Medical and Biological Engineering. I think we're all very fortunate to have the esteemed and deeply immersed in nanotechnology, Dr. Leary, with us today. Jim?
I want to thank Dr. Rothblatt for the very kind introduction and also for the pleasure in being here. As I told my wife before I came here, this is the first conference I've ever been where I'm in the middle. I'm not considered the extreme futurist. So it's been a pleasure to be able to say that. I'm going to talk about a lot of things here today, but I will try to draw a reference. A lot of things have been alluded to in earlier talks, so that they'll certainly make my job a lot easier. I came to Purdue about 2.5 years ago after 27 years as a professor of medicine, internal medicine, infectious diseases at the University of Texas, and earlier in pathology at the University of Rochester. So I come with a medical perspective, and now I'm in an engineering school with no medical school, so it's an interesting transition. However, there are many good things, and the thing that attracted me to Purdue was that they had just finished building a $300 million discovery park, bio-nanotechnology, which the Burke and Bindley centers were mentioned. And that gave me the exquisite capabilities for micro and nano fabrication, which is one of the reasons I'm there. I also have very excellent interactions and a cross-appointment between the engineering and veterinary schools, and we're going to do nanomedicine in animals before we do them in people. And I don't mean just in the nude mice that I'll show you, but actually curing animals. So I'll mention a little bit about that as we go along. Okay. I'm going to mention about programmable nanoparticles, and I'll say that in a very generic term because programming nanoparticles means controlling the steps of the interaction. So we're actually doing chemical programming, but it's very analogous to what you would get in computer software and doing that at nanoparticle level. So the first thing I want to say is that conventional medicine has been marvelously effective in the 20th century. We've extended the average human lifespan by 50%. We have not extended that yet, although I think we're going to see that in the next century. It's not science fiction, it's going to happen. And we've come along to realize that there are many, whoops, can I get back? Yeah, we go. We realize that there are also some startling limitations of conventional medicine, and we're starting to really hit the brick wall on that. And as such, it's been mentioned here how it's costing more than $1 billion to bring a new drug to market. That's clearly unsustainable, and in fact is an insane process. We have to just stop and say, you know, why are we doing this? So one of the things that's been held out as promise is so-called personalized or molecular medicine. And this depends obviously on genomic information about the individual, so that we're no longer guessing as to whether a particular medicine is going to be or procedure is going to be effective on a given patient. You don't like to think that you're a laboratory animal every time you go to the doctor, but you really are. And one of the interesting things that's come out of this is that we have, at this point in history, to the best of my knowledge, we have sequenced 2 individuals on this planet, Jim Watson and Craig Venter. And there is, I believe, a study in the UK where they're going to sequence about 1,500 people to look for single nucleotide polymorphisms, so-called SNPs, to see what the variation, it's the SNPs among us. We're about 99.9+% same DNA sequence as you and I, but we're all different. And if we can know those sequences, we can actually do something. But the interesting thing, so we'll do the SNP analysis first, but we'll have SNP chips here very, very soon. Particularly for certain diseases. But the thing that's fundamental about this is that I've sat on the study section for the last 3 years for NIH's program for the so-called $1,000 genome. And the purpose of this is to develop new technologies, a lot of them are nano-oriented, for very rapid sequencing and cheap sequencing of individual human genome sequences. And I'm not sure it's going to happen for $1,000, but I'm pretty darn sure it's going to happen for under $5,000. And that sounds like a lot of money. That's nothing. In the treatment of many diseases, we're spending hundreds of thousands of dollars per patient, and we have no idea whether the therapy is going to work. And in some cases, it's not only useless, but you realize as you treat a patient, you've done two things. You've lost time by going down the wrong route, and perhaps the critical time to keep them alive. And the second thing is, once you've treated a patient, that patient is no longer the original patient. They're a collection of all the things you've done to them, which in many cases invalidates another therapy. So we've got to start choosing better. So that's one of the processes. But this has been held out as the cure-all. And I would submit to you that it's extremely important, but we're still not going to solve the fundamental problem if we don't add to it. And I didn't say, originally I had an arrow here, but it really should be a plus because we're going to need both of those. Because we're going to need nanomedicine as single-cell medicine. And the reason for that is, as was said by Dr. Rothblatt very elegantly, as there are 3 laws of real estate, location, location, and location, there are 3 laws of drug development and delivery, and that is delivery, delivery, delivery. We have lots of fantastic drugs. We just can't get them to the right places in the right quantities without causing severe toxic side effects. And I would submit to you that this technology, nanomedicine, we can reduce 2 to 3 orders of magnitude the amount of drug that we give an individual going in with guided nanoparticle deliveries and more sophisticated things. And by doing that, automatically we're reducing toxicity and side effects. And as well, so one of the goals that I'm going to talk to you about is the way to be able to do that with guided nanoparticle delivery, and then also showing you how we can even put some fairly complex Boolean logic. It was mentioned about how we can actually start to put algorithms into nanoparticles. I'm not sure I've figured out how to put an ethical algorithm, but I will address that point because I'm actually trying to do that with one of my sequences here. And I didn't know that Dr. Bloomberg would be here today, and I'm not talking about our virus work. As it turns out, one of the first applications we did, actually before this grant, was actually doing hepatitis C virus work. And one of the marvelous applications of nanomedicine, since these are effectively the size of a virus, is that we actually made synthetic hepatitis C virus particles with the E2 protein on the outside of it, and the body treats it like it's a hepatitis C virus, which means it goes wherever hepatitis C virus does. Well, that gives you the possibility of developing marvelous antiviral therapies where they target and go in the body, and then you have a Trojan horse type therapeutic approach where you deliver something. And what we did was we were trying to deliver a ribozyme, which does a great job on killing virus in the test tube. It doesn't work at all in vivo because it doesn't last in bloodstream. But if you can protect it, then it's a pair of molecular scissors that can go in and actually cut the virus inside a living cell without killing the cell. Think how marvelous that would be. And I think this is a very general approach for antivirals. But not knowing that Dr. Bloomberg would be here today, I didn't think anybody else would be interested in that. So I didn't bring any of my slides for that. I'll be happy to talk with you later about that. What I did have in there, which is of interest to Dr. Bloomberg, is that I actually really got started in nanomedicine some 7 years ago through NASA, through the same program that he was affiliated with. And in fact, John Hines at NASA Ames was my program director under this. And it's interesting, this is one of those rare things that happens. The rumor has it that the director of NCI and the director of NASA went out to coffee together in Washington, D.C. I don't know if this story is true, but it sounds great. They went out to coffee and they decided to do something really wild. And so they came up with this program, which ended up being a biomolecular sensor program. And there were, in fact, 13 original groups, kind of like the original astronauts. Here we had 7 NASA-funded and 6 NCI-funded. I was one of the 7 NASA PIs. And interestingly enough, all 13 in these groups are still very, very active in the field. And it was around doing nanomedicine before NIH really started using the term. So it's fascinating, and it really got things off the ground. And and how I got involved in this was a 2001 conference by NASA, where I was invited to the conference, and some of the people in the program literally came up to me and said, "Jim, we have a problem trying to get astronauts to Mars and back. Frankly, they're going to get cooked with radiation. There's no way we can protect them. And can you try to come up with a nanomedicine solution to this?" And of course, in typical NASA fashion, they wanted an autonomous nanomedical device, launch and forget, and that would last in the body for 2 years or more. We don't know how to do that for a matter of days right now. But it was a great idea. And so we've actually got started with autonomous, and I'm going to talk about both autonomous and non-autonomous nanomedical systems. So it's a variety of things interesting things you can do. Well, the first thing that happened, you'll notice the date on this, is January 2002, was they had just finished funding this, and they wrote a very embarrassing article about me and my work and my collaborators. It went all over the internet, and it's still there, if you're interested, just Voyage of the Nanosurgeons, parodying Isaac Asimov's book on Fantastic Voyage. Now, the first thing you do when you see an article like this is you cringe and you hope your colleagues are never going to see it. Of course, that doesn't happen. I had more than 140 radio, television, and newspapers pursuing me for interviews, and I told them, "Go away, I haven't done anything yet. I just wrote a proposal." And of course, what they did was they went away. They're just starting to trickle back at this point. The one good thing I say about this is, it says in here, this isn't Hollywood, it's real science. So, in fact, I wanted to say we actually did most of what we said we were going to do. We have done all the pieces, all work, and now we're making integrated systems. But the interesting thing about this are, you always have to take these disadvantages and turn them around to advantages. And I said, if I can't escape this, I've got to find a good rejoinder. And so I said, I love Isaac Asimov as a science fiction writer. He's fantastic imagination, but he lacked imagination in one area for this Fantastic Voyage novel. He could not figure out how to put decision-making into that nano vehicle without shrinking people to nano size. And the interesting thing is that's the only science fiction that's in the story. The rest of it can be done scientifically. So what I'm doing now at Purdue, which has one of the top analytical chemistry groups in the world, is using chemistry to do decision making and making Boolean logic in a programmed series of steps for nanoparticles. So it's not going to be science fiction, it's going to be real science. Okay, and if you're interested in more of that, I'll put in a plug also that Purdue has the National Science Foundation Center for a lot of nano simulation and lectures. There's a lot of fascinating material on there. And I actually taught last fall a 21 lecture course. I did 17 of those and had 4 excellent colleagues contribute. And this is on there, and if you're interested, just go to NanoHub, www.nanohub.org, and there's lots of stuff on there, and I have a whole graduate level semester course on designing nanomedical devices. So in the vein of, this is not science fiction, we're doing this for serious, and we're going step by step through this. This is a slide to show you a couple of things. One is when people talk about implementing, actually making nanomedical devices, what kinds of things do you actually have to bring to bear? And we kind of talk about it in general terms about the different disciplines and things that you need. This shows you actually my laboratory, which I've now grown after a couple of years to a group of 9, including 6 graduate students. And these are the areas in my lab. Bioinstrumentation, a lot of it. And we have to do things, we have to measure things at the cell level because nanomedicine is single-cell medicine. So you have a lot of single-cell technologies that you'll recognize there, some that you won't. I don't have time to go through all of these. But, and then you have a lot of bio-nanotechnology issues And a lot of not only chemistry, but nanochemistry is very different. Things behave differently as nanomaterials. And so you find that many chemists don't have a lot of experience in that, and it really takes almost a hybrid type of chemist. We're doing a lot of sensor work because I'm trying to design some of the world's first single-cell feedback control release nanoparticles. And I'll show you that at the end. We've actually done that successfully. And you have things such as have been described here at this conference, such as atomic force microscopy. We're doing a lot of that, which is great on live cells because it can be done in a liquid environment. Transmission EMs, obviously. X-ray photoelectron spectroscopy, because if you talk about manufacturing these things, bio-nanomanufacturing is not something I think anybody in the world knows how to do at this point. There's a lot of things have to come together. I'm trying to put together groups at Purdue and grand consortiums here to try to approach the problem, but it's a problem much bigger than all of us. And then lastly, I've worked in all of these fields. I've worked in a lot of fields over the last 30 years of my career, including a lot of circulating cancer cells, rare cancer cell detection, what I'm trying to do right now is work in the area of regenerative nanomedicine, which I would argue is one step harder than a lot of the things that we've been describing today, because I'm trying to do something more than kill things. I'm trying to actually restore cells to at least a more normal state. So there's a lot of factors in here. Just wanted to show you that, and all these little plus signs you see here in each of these. You say, how do I do that with 9 people? Well, I have at any given time about 20 groups that I interact with. I'm a highly interactive person, and I see lots of possibilities for more interactions at this conference. So it's been, again, another delight to be here. One of the earlier things that I did in my career turns out to be helpful. When you're talking about putting nanoparticles in the human body or even in an animal, it turns out that what you really are always doing is the needle in the haystack. The targeted cell is always a rare cell, and it's always at frequencies typically of 1 cell in a million or below. And luckily, I spent 25 years of my career earlier than this actually into developing. I'm actually the original inventor of high-speed flow cytometry some 25 years ago. Took flow cytometers up to the level of 100,000 cells per second looking for 1 cell in a million. And people ask me, why are you doing this? And I said, well, gee, I think there's some interesting biology and medicine out there. And the thing is that if you don't have a technology, you don't ask the question. Now, it's very gratifying to see all kinds of questions being approached in rare cell biology, minimal residual disease monitoring, all kinds of things. And so I've done that. I've done a lot of work. A lot of other people have done work. We're literally able to walk away from a cell sorter with a single cell. We actually have done gene sequencing at single cells to see a single base pair deletion in a single cell. We've shown that that can be done. In this interesting paper, if you want to follow any of that describes all this right here, and it'll be in the slides that you'll have access to. The other thing you can do is turn this around, because a lot of people have been thinking about putting antibodies on nanoparticles, and we certainly have been doing some of that also. But really, antibodies are too big for nanoparticles. They're too big, they're too immunogenic, it's very hard to make antibodies which don't have all kinds of side reactions and other problems. And what we'd really like to do is use much smaller molecules. And that leads us to a lot of things such as this is doing, if you have a high-speed cell sorter like this, then you can start approaching the problem a little differently, like Mother Nature does. And that is, you approach it combinatorially. And so one of the first things we did a number of years back, about 5 or 6 years ago, we started doing combinatorial chemical library screening. And we were doing this particularly with aptamer sequences. These are thioaptamers with the thiol groups linking on the backbone, double-stranded DNA, and they actually have recognition just like an antibody does. But they have some neat properties. One is they can be manufactured very easily. You can manufacture, this particular one that I show you here is 10 million compounds that we analyzed one by one in an afternoon. We had one sequence on each of these small beads, and we did this by high-speed flow cytometry, single bead sorting, and then we could sequence. And you pick out the ones that works, just like Mother Nature does. Rather than trying to design it, you say, I don't even begin to know how to design something like that. I'm not smart enough, so why don't I just pick the winners? Okay, it's sort of like picking the horse race after the horse has crossed the finish line. You know you're going to win, even if you couldn't have predicted it before the race started. So we started doing that. And you can do the same kind of thing with peptide libraries. And in fact, I'm going to show you some peptide results, not actually libraries we screened, but that other people did, because once you have the sequence, then anybody can easily manufacture that. So, and then what I'm not going to mention, but if people are interested here, as I said, I was the original inventor of high-speed flow cytometry and sorting, and what I'm trying to do is build the world's fastest biomems cell sorter that will go, that'll be the size, as I say, of my cell sorter the size of my PDA with the chips that go in and out. And it's going to happen. It's been a little tough slogging through that work, but we're well on our way here toward creating chips, disposable chips, and portable flow cytometers and cell sorters. Okay, so why do we want to make a programmable nanoparticle, and how do we do that? Well, the first thing that we do is we recognize that programming is controlling the sequence of events. It doesn't matter how you do it, okay? And one of the ways we can do that is to make a multi-layered structure, which then only the outermost layers control that given event, and that you have to peel that layer off and go to the next event, and the next event, and the next event. And the consequence of that is that you can actually do fairly complicated Boolean logic with that. You can certainly do ands. If you're clever, you can do ands and ors, and even nots in there. So you can make a very complicated program in this structure. And as you go down through this shows you where the outermost layer has been designed to hook on to some cell surface ligand or receptor. It may strip off that layer as it goes through the cell membrane. It may then retarget. And why do you need to retarget? Well, it turns out that we haven't talked about that today, but in scaling terms, a nanoparticle is about 1/1,000,000th to 1/1,000,000,000th the volume of a human cell. So it's a huge place in there. And to give normal drug delivery, if you're going to deliver some drug, how many drugs can you dilute 1/1,000,000th fold and still have them be effective? So what that means is, can you do that? And the answer is yes, you can. But if you retarget, you get inside here and you retarget, and we spent a lot of time doing, you can also use amino acids for retargeting, amino acid sequences for retargeting inside a living cell, and actually put it to a particular organelle or a three-dimensional location within a cell. And so we've done a lot of confocal imaging and shown that we can actually target to specific regions inside a cell. So I'm going to run through a very quick example, and this is from a recent thesis of one of my students, just going very, very rapidly, showing you that it can be done. This is a breast cancer cell targeting, first of all in vitro and then in animals, where we had human SKBR3, it's a highly metastatic breast cancer cell line. And what we did was we used a peptide that's specific for SKBR3 cells. In fact, it even distinguishes between other breast cancer cell types. And this was published by this group. And we just simply took the sequence and manufactured that and then did and this again, as in earlier talks, is a relatively simple 7 amino acid sequence. And yet it does marvelous things. It actually does the equivalent of 2 of those layers. It does the cell surface targeting, and it actually promotes entry across the cell membrane. So it does. So I would submit to you that multi-layered nanoparticle is actually a paradigm for all nanoparticle systems. Most people don't think of it in those terms, and when I teach my course, I always say, whether they call it that or not, that's what it is. Some people have a single layer, some people have 2 layers. It turns out by the time you get to 3 or 4 layers, you can do some very sophisticated things. So what we did was we decided to do some very rapid prototyping using quantum dots, which we've talked about here earlier today, because we can at least see them optically, or at least we can sort of see them optically, and I'll point out to you why. So here's an example of a commercially available now from Invitrogen of a quantum dot with a cadmium selenium with a zinc sulfide core. And typically these things with the biocompatible layer are about on the order of 12 to 14 nanometers across. So they're pretty small. And then you can start tagging a few other things to the surface and start putting some peptide guiding of this process. And so one of the things in particular we did was we took this targeting sequence against the SKBR3 ligands on the surface of these cells, and we conjugated them, and I won't go through the chemistry here, but it's pretty straightforward chemistry, not very sophisticated, and we confirmed that we indeed had it successfully attached and in the right orientation so that it was able to recognize the cells. And just putting this in perspective, remembering that this is about 1,000 times bigger in diameter than these quantum dots. So we're talking a billion-fold bigger in terms of volume. So just keeping that in mind here. And what we did was we actually compared two things. One is we took quantum dots with a universal peptide, and they call these, in commercial parlance, Q-tracker quantum dots from Invitrogen is the ones that we actually used. And, and what we did was we compared that. So that will essentially bind onto almost any cell type, and it'll go in, but it'll stay in the cytoplasm of virtually all cells. So you can put it in there and use them as a tracking device. This particular sequence goes into SKBR3 cells only. So what we did was, and you can't see this image very well, but I'm not going to worry about it right now. Just so we did a lot of confocal imaging on this, and we showed that it goes inside, the SKBR3 goes inside, and it stays in the cytoplasm, and it also doesn't label MCF7 cells, which is actually what we use, another human breast cancer cell line that we used for a control. And we did a lot of experiments with mixed cell populations, and as you can imagine, you do this by image analysis, you can do this by flow cytometry, so you can actually look down, can you really detect something at 1 cell in a million? And you can do those kinds of experiments if you have the appropriate technologies such as we have. And just very, very quickly, what does this mean? So with the universal peptide, everything labels as expected. This is a DAPI stained nuclei, and you see that every cell labels very nicely. And when you do the experimental control with that particular sequence against the SKBR3 cells, you'll see the MCF7 cells, you'll always get, and this one that I show here in this sequence right here, you might say, well, gee, that's pretty bad because it's 1 out of 5. No, that was 1 on the slide. So it was very, very sensitive. And we're talking, scanning across, millions of cells. And that was, so it's not a representative sampling, that's the one that we picked out. And this one is, it's not 100%, but it's darn close to that on picking out the SKBR3 cells. Now, this one isn't a very interesting slide, but I want to take a couple of take-home messages from it. When people do a lot of toxicology assays, one of the problems is people are hoping for a negative result. You kind of tend to get the result that you're hoping for if you look for it in that way. And it's very easy to get a negative result because, in fact, a lot of these nanomaterials don't immediately kill cells. And you'll see a lot of people do assays like MTT assays, which are pretty gross killing assays, or they do live dead cell assays, trypan blue, PI exclusion, and so on. You can get into more subtle things like tunnel assays, which is a late apoptosis or programmed cell death assay. You can do earlier ones, which are presumably you're getting more sensitive with the Nexin-5, which is a molecule where you have the phosphatidylserine externalizes from the inner part of the membrane to the outer part during early apoptosis when a cell is pretty much going to die. It's just a question of how long. And then an Nexin-5 is a molecule which binds to the phosphatidylserine. So we did all of those assays, and like a lot of people in the literature, we found that nanomaterials, at least a lot of the ones that we've been using, not only just this, we did a lot of work with other materials, are not very, surprisingly not very toxic in nanoform. However, you very quickly, when you get to something like a semiconductor material like a quantum dot, you say, what happens when you hit it with light? So we did a lot of experiments with very high dose UV light to try to break down the surface properties, because a lot of these things, if you go into one cell type, you see no toxicity. These are the papers that you get into science, and then you say, that's great, but it only works on that cell type, and it's lethal to these other cell types. So you got to be very careful. And so what we found was that, in fact, with the cell types we were using, we could really bombard them with UV, and we didn't get much, mainly because the biocoating stays on. But if you have cells that strip the biocoating, and some do, then you can get much different toxicity values. But one of the things that we did do with these experiments, we looked at the cells, and being reasonably good cell biologists, we said, all these assays are coming out negative, but those cells are not happy cells. They're not happy campers in there. Why is that true? And so we went in there a little bit further, and we discovered one of the things that happens, as you know, is oxidative stress. When cells are stressed, they may or may not die, but they're kind of at a trigger point. They're either going to get better and repair themselves, or they're going to die. And we, in fact, did a lot of studies with that. And what we found was with these quantum dots that, in fact, the Q tracker ones that everybody uses for tracking actually produce immense amount. This is an assay, dihydroethidium assay, very simple assay. This is a hydrogen peroxide control. Everything should be positive. So here's a positive control, here's the negative background, and here's what happens with quantum dots that everybody says doesn't harm the cells at all. These cells are highly stressed. Okay, they're about to have a nervous breakdown. Okay, whether they go into apoptosis or not depends on whether they have the appropriate things to recover. So the answer is there's a lot of things, and you only, I guess it was because I dug harder. My graduate student, many experiments coming up with negative results, and I kept pushing her in her thesis, said, no, something's still wrong there. So I was looking for the result that most people don't want to find, and I found it. So you got to be careful. It doesn't say that these cells may not recover, but be aware that they can be very stressed. So one of the things we did was we immediately took that experiment in vivo. And the logical thing to do, it's not a real, what I call a real experiment with an animal with an intact immune system, but you couldn't do this with human cells. So what we did was we took a nude mouse, which has been discussed earlier today, and that's a mouse that's not only nude in terms of hair, but nude in terms of not having an immune system, so that you can actually transplant human cells. And typically what you do is you transplant, in this case, the SKBR3 human breast cancer cells into the flank region in the back. And what will happen is an interesting thing, is that in most cases, tumors don't grow unless blood vessels grow and so-called vascularize and feed the tumor. So what we did was we actually did all those experiments, and we collected tumors. We did a lot of nude mouse experiments here, and actually found the tumors that had vascularized and grown, and then did, in typical pathology section, did sections. Lots of sections, over 700 sections. And again, my student's thesis reflects why we don't want to do that in general. It's a tough way to go. So I'll discuss in a better way here at the end of the talk. So one of the things we did, just to take you very rapidly through that. So here's the control. This is with a high sensitivity black and white camera. So I'm not showing this in color, but in fact, here's the sort of normal histology. Here's where we actually did a positive control. We took the quantum dots and we went directly into the tumor that was clearly visible. So we could say, what does it look like when it's positive? And these are the quantum dots in here, the bright things. And then this is the really interesting experiment, because we've heard about photodynamic therapy, for example, just an earlier speaker. Photodynamic therapy works because the dye goes into the tumor region because the vasculature around tumors tends to be leaky. Okay, so it just leaks in there, and that's part of the therapeutic treatment. Well, if you look out in the world's literature, about over 80%, closer to 90% of the world's literature with nanoparticles is totally unguided nanoparticles that are taking advantage of leaky vasculature. So if you do that, you say, if I put enough nanoparticles in, they will indeed get to the tumor. But the answer is you got to put a lot of them in there. So we took the opposite tack and said, what if we use peptides to guide those nanoparticles? Let's see if we can go down at least an order of magnitude lower than anybody else in the world has done. And the answer is we did that, and that's right here. This right here, and you can see the individual tumors inside the other cell types in here. So we're getting a collection of each of these. And when you see quantum dots at the fluorescence and light microscopy level, one of these dots does not represent an individual quantum dot. That represents thousands of quantum dots. And in fact, we went further. We were pretty obsessive about this. We actually counted the dilution factor. So we're down at less than 10 nanomolar quantum dots in the animals. We're actually counting heartbeats and circulation times and looking at accumulation rates as a function of that. Well, it turns out, well, it's kind of important because the probability in a given circulation is some fraction of 1%. So it takes a certain amount of time, but you're also, as they're guiding to the tumor cells, you're also screening them out. You're filtering them out, as has been talked about earlier with regard to the kidneys and the zeta potential and so on, that you have to be very careful how you design these things. And in fact, so you have this competition where you're putting things in and they're getting filtered out. And what you find out is that actually this levels off. It's detectable at 1 hour after putting it into the tail vein of the mouse. And then it has to find the tumor. And that's a pretty tough job to do that, to go through the circulation and find it and get into the tissue. After 1 hour, you light these up. And after about 4 hours to 8 hours, you reach pretty much saturation, because you're sitting there taking the ones out, which leads to another problem, saying we took an order of magnitude less than anybody else, but if we could increase the circulation time, and that's the key, if we can increase the circulation time and increase the targeting, we may be able to go 2 to 3 order magnitude reduction in the amount of drug. Now think what that means in terms of FDA approval, think what it means in terms of toxic side effects. I mean, we are flooding our bodies with drugs right now compared to what we're talking about at the concentrations that it's possible to achieve. So this is, I think, represents a pretty significant advance. And this is just reminding you that these are pretty big things. And when we actually, we got compulsive again. We said, how many nanoparticles do we put into each mouse? And we found out that we were, even at these kinds of labeling, we were putting in about 10 to the 13th power nanoparticles per mouse. And we were getting labeling and uptake on the order of about 10 to the 11th nanoparticles uptake in the tissue, and relatively specific, which is pretty good. That's still only 1%. We're still throwing away 99%. So, but that's just giving you some real numbers, because we actually went through and crunched all the numbers on this. Now, let me get to the very last part of the talk, because I think this is the interesting part. So we're not planning on doing quantum dots for in vivo, and particularly for human, because there are toxicity questions, although if we can get the amount down, we may be able to do something else. But what we are doing is we're actually trying to do something a little bit different, because what I didn't mention to you was on this dosing problem, so far we've been worried about killing things. What if you had to get exactly the right dose, not only the right dose for the person, but if nanomedicine is not dosing the person or the organisms, it's dosing at single cell level. So you've got to get the right dose cell by cell. How in the world can you do that? Well, one of the things you can do, and this is an interesting thing that you've all heard about biomimicry, that this basically says nature inspired. One of the interesting things in science is that when you know something can be done, it's amazing how fast it gets done. We all hang back. We all say, oh, it can't be done, it can't be done. I always get on my students saying, don't tell me 10 different ways why it can't be done. Make your life easier. Just come to me with one reason how it can be. We talk ourselves out of a lot of things. So the beauty of this, when people said to me I was going to set out and try to do something like making effectively a synthetic virus delivery package, I said, gee, that sounds terribly hard. And I said, oh, no. I'm absolutely 100% certain this will work. I may not be the first person to do it, but I'm positive this can be done. They said, how are you so sure? I said, well, nature did it millions of years ago already. So I'm not sure nature should get the patent on this. But I do have one pending. Anyway, so we're trying to make let me go back one here. So very quickly, so what we're trying to do is make effectively a synthetic virus that has no ability to make copies of itself. So that's my ethical decision. We're not going to make any self-replicating ones. And the other thing is, if we can basically control this process well enough so that we can make what I call a nano Lego block assembly, where we actually make this so that you can easily swap molecules, targeting molecules or therapeutic molecules, in and out of this structure. Why is that important? It's because the FDA, and I gave an invited talk to the FDA last year, they're trying to figure out what to do with nano drug delivery devices. So what I'm trying to tell them, first of all, let's treat this as an extension of directed therapy. We already know we have Rituxan, we have similar therapies out there. We could deliver those same therapies, first of all. It's really a similar technology. So right now, the FDA doesn't have a policy on this, and it would be like if the FDA were approving your car, and they had it all approved, and then you said, oops, I have to change the windshield wiper blade. They'd say, oh, we're terribly sorry. We have to start all over again, reapproving your car. Now, this clearly isn't going to work. They know that. They know that they're going to destroy nanomedicine if they require that thing. But what we're going to have to do is come up with a way of certifying types of vehicles that we swap molecules in and out and have some predictability, and because we're using such a low dose, that the toxicity effects are hopefully going to be minimal anyway, so that we can basically make nano drug delivery systems that we can rapidly make a platform technology. So I was arguing pretty strongly for that. And so the challenge of drug delivery, we talked about delivery, delivery, and delivery. Well, the second part of that is dose, dose, and dose, okay? And particularly if you're going to do regenerative medicine, how do you get exactly the right dose? Well, the easiest thing is listen to the cell. Let the cell tell you what the dose is. And how do we do that? Well, it turns out there's a simple way to do that. First of all, just show you, reminding you, this is the kind of structure. And instead of putting a drug in here, what we're going to do is we're actually going to use a contrast agent. We're doing a lot of MRI now and doing ferric oxide cores, which are already, core material already approved for human use. And it turns out that superparamagnetic ferric oxide cores, about 30 nanometers across, even in a 3 Tesla human MRI machine, which many of you have been in, can actually resolve. Normally, you can only resolve about a ½ centimeter or greater size tumor in a 3 Tesla magnet. We're getting down to about 1 millimeter with this. This is with a non-optimized. I've got a student, new PhD student, who wants to really do some, we're going to do some, I think, some fun things redesigning, making new contrast agents for high resolution and perhaps even multiplex MRI, which would have tremendous power. And so what we do is we build this thing layer by layer. So here's the core material. We then put in the surrounding layer therapeutic genes. Now, a lot of people, we've talked about, Dr. Plumberg talked about how hepatitis B integrates its DNA into the genome of the cell. I don't think we want to go there, particularly with some of the history of gene therapy so far. But what if we can get people to accept that transient gene therapy is just that? It's just another drug. And why do we want transient gene therapy? It's because why deliver a drug when the drug components are already there? The cell is full of DNA and RNA precursors. All it needs is the template. So we have everything we need inside except for organizing that. So one of the things that we're doing then is we're delivering in this therapeutic gene an actual template to manufacture. So why deliver a drug when it's got everything there? We can take these off layer by layer, cellular targeting, intracellular targeting, therapeutic genes. We can make all of this biodegradable. And then what we can do is we can actually take that multi-layered nanoparticle, deliver it with the gene manufacturing machinery. How do we do that? We use things that we already know that viruses know. Viruses have transactivating agents that can actually use the host cell machinery to make particular things that it wants to make. So we'll use the same types of things in here. The neat thing we'll do with this, which is incredible, I tell all my Purdue engineers, you know, no self-respecting engineer would ever design a system that didn't have a feedback loop in it. You want to have something that's not a runaway and not a control. So why not put an upstream molecular biosensor switch and have this in a normally off state so nothing is happening? So guess what that also means? Not only do you have the Boolean logic in this, but you have error checking. If you go inside and you say, whoops, I got in the wrong cell, I don't see what I'm looking for, it doesn't do anything. So as part of this then, the interesting thing is you start seeing nanomedicine is not, I always tell people nanomedicine is not nanomedicine because it's nano-sized. That's really the smallest part of it. It's nano for 2 reasons. One is that it is bottoms-up medicine, okay, it's cell by cell. And the second thing is that medicine as it is right now, as we've talked earlier today, has been based on symptoms. All of this is going to be pre-symptomatic. The line between prevention and disease, all the symptoms we call early right now are going to be considered late. Within 10 years, I would predict. So a lot of these are going to be molecular tests. And what we're going to do is we're going to be looking for the presence of specific molecules, either disease-specific molecules or surrogates. We don't have to be perfect to get it right. But if we can find a disease-related molecule that we can then design a molecular biosensor, and you can design molecular biosensors against almost anything, you can design a switch which this molecule, when it comes over and binds to this, will then turn this machinery on. And when you keep, and then the purpose of this machinery is to effectively inactivate, change the conformation, otherwise deactivate this molecule. When there aren't any more of these to come back and on the switch, then the switch goes off. And how do we know we get the right dose? Because cell by cell, this goes only as long as it needs to be able to reduce the target molecule to the point. So complete feedback loop. Now, this sounds very fantastic science fiction. I assure you it isn't. We've already done this. Let me show you an example. We've done this and actually published this. We've made, first of all, we've done, this shows one of the first papers, my former graduate student, part of his PhD thesis, and now with Jerry Ludy's group at Johns Hopkins. We've made these multi-layered nanoparticles. And one of the first things we did was say, can you read a gene sequence off it? Okay, it's not as simple as it sounds. Well, the answer is, if you tether it properly to in this case, a superparamagnetic nanoparticle, you can get very, very high levels of expression. And we did a lot of experiments using green fluorescent protein reporter molecules that would light up as you read the gene sequence successfully. So the answer is, yeah, you can do this very efficiently. And then the next thing you can do, you can go one up in that. Let me show you. There's two important results here. The first one is that the case where you take a cytomegalovirus promoter on a DS red, which is this red material, it's another reporter gene material, and then you use the transactivating sequences and so on to be able to get it to describe. So this was done with these nanoparticles, which went in and then just made copies of this fluorescent protein, which could have been the therapeutic gene. We just had it sitting there lighting it up. And so that one was sort of like, okay, we can turn it on, we can manufacture it, but the real subtlety is in the green one here, where in this particular one, what we took was we made a molecular biosensor switch against oxidative stress. And this is one we did not design. It was, again, a published sequence, molecular biosensor sequence. We hooked that upstream to a promoter region for enhanced green fluorescent protein, and we actually went inside, we stressed the cells, and we actually had the green fluorescent protein reading this off in response to cellular stress. Now, what I haven't shown you is here we've gone one step further. We've actually, because oxidative stress causes DNA damage, we've actually gone in and put a DNA repair enzyme on and repaired the DNA. So pretty cool. And that was actually for the NASA project that we were trying to do. Could you repair the DNA in radiation-damaged blood cells of astronauts? And we did an accelerated DNA repair introducing a second alternative pathway on one of the radiation-damaged. So there's I could give a whole lecture on that, but I won't. So luckily for you, I'm almost done here. So where are we going with this? Well, the interesting thing is that other countries are spending a lot more money than we are on this. And I now head up I'm sort of embarrassed to say that it's if we want decent funding for our research, we kind of have to go outside the U.S. these days to get it. And one of the things, both for the innovativeness and also just plain dollars. And so I've teamed up with a group at the Korean Institute for Science and Technology in Seoul. And we have now a 9-year relationship funded by the Korean government. Instantly put $4.5 million into it just to start things up. They're thinking 10-year plans. I just came back from India. We're trying to do a similar thing in India, where they think in 10-year plans. And you and I know in the U.S. these days we don't know what our budget is next year, which is a really hard way to do science. So but what I want to say, the other thing on this is that we're combining this with molecular imaging. You've seen examples of earlier talks of near-infrared probes and optical imaging, but I would submit to you that it's very hard. While we can squish a mouse down on a microscope slide, most of us are a little broader than that. So one of the logical ways, I thought it was very interesting, the X-rays. I want to talk with you more about that with this. That's a fascinating alternative. But we're going with magnetic resonance imaging, making these cores, and we're doing what we call theragnostics. And that's a new word you're going to see a lot of, theragnostics, which is simultaneous diagnostics and therapeutics. And with the beauty of this is that you get location, you can follow a patient or an animal serially in time as you treat it. You can watch tumor shrinkage, you can watch disease progression or lack thereof. So it's a way of getting, and we all know it's easier doing an intra-study than an in-between study, so between animals or people. And we don't have to do hundreds of sections of histology. And lastly, I just want to credit, as always, I get the fun part of traveling to meetings and giving talks, but a lot of this is by the very hard work of students. In this particular, you saw a lot of the quantum dot work of my Emily Haglin, who just defended her thesis. The new ferric oxide nanoparticles is being done by Mary Margaret Seal and Jaehong Key. And we have very excellent collaborators in the doing animal experiments and MRI imaging and chemistry. So it's with the, as I said, these are the people backing us up. There's always a lot of backup here for any kind of work like this. It's very complicated work. That's why you need a lot of collaborators, and I tried to show you some of the components. We do an awful lot of different types of things here, and no way we can do them all ourselves. And lastly, if you're interested, there are some recent publications and a pending patent on these kinds of ideas. So with that, I'll be happy to answer questions.